Hello everyone. So today we are going to talk about polyadenylation. So polyadenylation is basically the addition of a poly-A tail to the 3' end of an mRNA molecule. We have previously discussed that these processes, the post-transcriptional modifications, which include 5' capping, 3' polyadenylation, and splicing of the introns, these processes take place inside the nucleus. So it happens that this is an mRNA molecule. As soon as it comes out of this transcriptional initiation complex, so which initiates transcription, and the formation of the synthesis of the mRNA molecule takes place, it comes out at the 5' end. And as soon as it comes out of this complex, the 5' end is immediately capped, what we have discussed previously. But later the 3' end is cleaved by a ribonuclease, and here it has got a poly-A site. Poly-A site is the region where a poly-A tail is added to the 3' end of the mRNA molecule. So this process takes place inside the nucleus. How does this take place? We will study this later. But what we need to talk about is two other types of polyadenylations. One is cytoplasmic polyadenylation, and the second one is alternative polyadenylation. So as far as cytoplasmic polyadenylation is concerned, till today we have been studying that this polyadenylation takes place inside the nucleus. But in certain scenarios it also takes place inside the cytoplasm, and this is especially true in germ cells and in nerve cells. So in embryos, during the developmental process, the maternal mRNA molecules they go through this polyadenylation process inside the cytoplasm, and generally they have the length of the poly-A tails is less than 20 nucleotides, and inside the cytoplasm an addition of roughly 80 to 150 nucleotides is made inside the cytoplasm in these germ cells. So generally it takes place because the transcription does not start till 2-cell stage in case of mouse embryos, and till 4-cell stage in human embryos. So these mRNA molecules which are already transcribed, which are the mRNA molecules which have already been synthesized, and these are maternal. The origin of the mRNA molecules is maternal because they reside inside the ovum. So these are maternal mRNA molecules, and the cytoplasmic polyadenylation takes place because so that they can be translated till the transcription is initiated, because the transcription is not initiated until it reaches a 2-cell stage in case of mouse embryos, and a 4-cell stage in case of human embryos. So this is about the germ cells, and then we talk about the nerve cells. Inside the nerve cells, in brain cells, the cytoplasmic polyadenylation, it is active generally during learning, and is supposed to strengthen the transfer of signals from one nerve cell to another nerve cell. Here's an example. So what we see here, this is a neuron, and these are the dendrites of the neuron, and here's the cell body of a neuron. And what we see here, this is let's start from here. So this is a DNA. So generally what happens, a DNA leads to the synthesis of an mRNA molecule. This mRNA molecule is then exported out of the nucleus onto the rough endoplasmic reticulum, which then leads to the synthesis of the protein. But in certain scenarios this tail is pretty short, and when this tail is short, so the tail also helps in translation process. We will study this later, how does it play a role in translation process. So there are certain proteins which are associated with this poly-A tail, and they help in the translation process. So this is an mRNA molecule. If it is not meant to be translated, so this is generally kept repressed by the binding of protein and mRNA binding proteins. So these mRNA binding proteins, they keep this RNA molecule repressed, and later this when this molecule is this mRNA is exported out into the cytoplasm. This is generally carried by the kinesin motor proteins and brought to the dendritic spines. And inside the dendritic spines, we know that these are cytoplasmic extensions, and these extensions are basically they bulge out of the main cell body because of the presence of these actin molecules. So these actin proteins, they push the cytoplasm towards the periphery, which leads to the formation of these dendritic spines. So these motor proteins, the kinesin proteins, along the microtubules and then along the actin filaments, the myosin motor proteins. So these are two types of motor proteins. Initially, the kinesin motor proteins, which take the mRNA molecule along the microtubules, and then they hand it over to the myosin motor proteins, which carry this mRNA molecule along the actin filaments, and they bring it closer to the dendritic spine. And during this process of traveling all the way from the nucleus to the dendritic spine, the length of the poly-A tail is increased, and this is where it becomes capable of being translated, and this is translated into a protein in the dendritic spine. So this is the second phenomenon this is the second place where polyadenylation can take place. So it can take place both inside the nucleus as well as inside the cytoplasm. It is not common for the cytoplasmic polyadenylation to take place, but it does take place in certain cell types, like in germ cells or inside our nerve cells. And inside the nerve cells, it is inside the germ cells. It is known to support the growth of the cells because there is no transcription taking place in the initial multiplication phase of the cells. And inside the nerve cells, it is meant it is meant to play a role in long-term potentiation. It perhaps play a role in long-term memory. So these are two types of polyadenylation: nuclear polyadenylation and cytoplasmic polyadenylation, depending upon the location where this polyadenylation takes place. Let's talk about alternative polyadenylation. What is alternative polyadenylation? Is the genes may contain more than one poly-A sites. So poly-A site is the region where cleavage takes place and then a poly-A tail is added. So in example A, we have a normal usual scenario where we have an mRNA molecule, and so this contains a poly-A site here. So this is a cap. So we have a start site and a stop site for the translation, and then we have the 3'. So the blue one here, this is the 3' UTR, the 3' untranslated region, and the green one here is the 5' untranslated region, the darker green color. So this is the 3' UTR, and inside the 3' UTR we have a poly-A site. So what happens that this is a mature mRNA. A mature mRNA is synthesized where introns are removed. So these are the introns, the green lines. They're removed. So capping, splicing of the mRNA, the exons, and then you have this 3' UTR. So when this is translated, it is translated into a protein product. So this is the normal scenario. Let's see what happens in case of scenario B, that this gene now also contains an additional poly-A site. So rather than one poly-A site, this contains two poly-A sites. So in this case, what is going to happen is that we are going to have two different transcripts. This is one mRNA molecule, this is the second mRNA molecule being synthesized from the same gene. So this gene is capable of producing mRNA molecules with two different lengths. So if a poly-A tail is added here, then this is going to have a longer 3' UTR. And if a poly-A tail is added here, so it means the mRNA molecule is cleaved here and then poly-A tail is added. So this is going to have a shorter 3' UTR. But because it did not affect the coding sequence, both the transcripts are going to produce the same protein product. What would be the effect of having a shorter or a longer 3' UTR is not that it is going to produce a different protein product, rather it can affect, for example, the lifespan of the mRNA molecule inside the cytoplasm, because 3' UTR is the region where lots of different proteins bind, and it is also a region where the proteins which are involved in mRNA decay bind. So the expression profile of the gene may get affected, but the protein product is not affected. So in another scenario, we have another scenario where the protein product can get affected. So in this case, one gene can code for more than one type of protein. For example, in situation C, what we have is that this is the normal poly-A site, and the second one, what we also call the proximal poly-A site, this resides inside one of the exons. So when it resides inside one of the exons, it means the mRNA molecule will be cleaved here and poly-A tail is added at this point. So this is going to synthesize an mRNA molecule where we do not even have a 3' UTR. So it is going to produce a truncated protein or a different type of protein, because this is inside the exon. So in another scenario, we have this inside one of the introns. So this mRNA molecule would be shorter comparative to the normal molecule, but it also contains a short strand of the a short sequence from the intron as well. And these two things happen because changes in the polyadenylation site are coupled to the RNA splicing. So if you change the site for the addition of the poly-A tail, you may also change the RNA splicing sites. Means the mRNA molecule can be cleaved here, it can be cleaved here, it can be cleaved here, or it can be cleaved here. So it can have an impact on the cleavage of the mRNA molecule where it is cleaved, and it can also affect the length of the mRNA molecule. And because you produce mRNA molecules with different lengths, you can also have protein products which are different from their usual protein products. And this is also why one gene can code for more than one protein. So in both the scenarios, what we have learned that if a poly-A tail is added at different points inside the 3' UTR, because this is an untranslated region, this is not going to get translated into a protein. This is not going to have any impact on the protein itself. Rather, it can have an impact on the expression profile, targeting, translocation of the protein, and things like that. But the protein product remains the same. While in another scenario where the coding sequence or the non-coding sequence, that are introns, they're affected because the proximal poly-A site lies somewhere within the coding sequence of the mRNA molecule, or lies towards the 5' end or upstream from the transcription stop site. So this can lead to the formation of truncated mRNA molecules, and because the mRNA molecules become truncated, they can also lead to the formation of different proteins. So that's all about one other thing. As I have told previously, that the length of the poly-A tail can also have an effect on the expression profile of the gene itself. So here's one example. So we know that microRNAs or siRNA molecules, this here it is written siRNA, but generally we have these microRNA molecules inside the cell, and these microRNA molecules are involved in regulation of gene expression. So if an mRNA molecule has been synthesized by a gene but needs not to be translated, it can be degraded by the binding of a microRNA molecule to it. So this leads to the formation of a double-stranded RNA molecule, which is immediately degraded. So if you have a complementarity with an siRNA or microRNA molecule in the 3' UTR, so this is polyadenylation, this is the 3' UTR. A microRNA molecule or an siRNA molecule can bind to it, which would lead to the formation of a double-stranded RNA molecule, and this double-stranded RNA molecule cannot survive and would immediately be degraded by the cell. Hence, the gene cannot get expressed. But the difference it can make, as far as the length of the of the 3' UTR is concerned, because of the alternative polyadenylation sites, the same molecule can either have a full complementarity or a partial complementarity, depending upon the length of the 3' UTR. So if this was a full length or normal length or whatever molecule with the longer poly tail, so we can have a full complementarity of the microRNA or siRNA molecule, and this can completely silence the gene. While in case of partial complementarity, this may not completely silence the gene, but it can downregulate the expression profile of the gene itself. So we have learned now that we have this nuclear polyadenylation, cytoplasmic polyadenylation, and alternative polyadenylation. Let's talk about the process of polyadenylation. How does this take place? So we have discussed this, that in an mRNA molecule we have a 5' cap, and we have polyadenylation at the 3' end. So how does this take place? At the 3' UTR we have a poly-A site. So poly-A site is the sequence or the region on the mRNA molecule that would be cleaved from the pre-mRNA molecule. This portion of the pre-mRNA molecule will be degraded, and a poly-A site, a poly-A tail will be added to the poly-A site, and that would lead to the formation of a mature mRNA molecule, where you have a 5' cap, all the coding sequences where introns have been removed, and addition of a poly-A site. So this is a pre-mRNA molecule. So this still contains this 3' region, which needs to be cleaved at the poly-A site, and poly-A tail is to be added at the poly-A site. So do not get confused with poly-A site and poly-A signals. Poly-A signals are two sequences. Basically, we have two poly-A signals. One is downstream of the poly-A site, and the other one is upstream of the poly-A site. So this is downstream poly-A signal, and this is upstream poly-A signal. This is also known as GU-rich region or U-rich region. So these two are the sequences where 4 factors can bind. These are 4 nuclear factors. They are found inside the nucleus. So you have this pre-mRNA molecule inside the nucleus where a cap has been added, and it contains 2 poly-A signals and 1 poly-A site. To these 2 poly-A signals, these 4 nuclear factors can bind. So the first one is a cleavage and polyadenylation specificity factor, CPSF. CPSF recognizes the upstream poly-A sequence and binds to it. This is then followed by the binding of cleavage stimulatory factor to the downstream poly-A signal. So CPSF binds to the, CPSF, cleavage and polyadenylation specificity factor. So this binds to the upstream poly-A sequence, and cleavage stimulatory factor binds to the downstream downstream poly-A signal. This is then this cleavage stimulatory factor also interacts on the other end. At one end it binds to the downstream poly-A signal, and at the other end it also interacts with the CPSF, which was bound to the upstream poly-A signal. And this creates this binding between these 2 factors, leads to the formation of a loop. This loop is then bound by cleavage factor 1 and cleavage factor 2. So the job of these 2 factors is to stabilize this complex. After this complex is stabilized, another enzyme, what we call PAP or poly-A polymerase, the PAP then is recruited and binds to this complex. So this PAP, the job of the PAP is to cleave the mRNA molecule at the poly A site, and this generally cleaves it roughly 10 to 35 nucleotides upstream from downstream or towards the 3' end from the upstream poly A signal. So this poly A signal, so 10 to 35 nucleotides towards the 3' end. This is the cleavage site where poly A polymerase would cleave this mRNA molecule. So here it is. So this is being hydrolyzed. So OH means this is being hydrolyzed at the 3' end, and this leads to the cleavage of the entire section. So all these like the cleavage factor 1, cleavage factor 2, cleavage stimulatory factor, so these three factors along with this short strand of 3' end of mRNA molecule, this gets dissociated from the main mRNA molecule. So after getting dissociated, this gets degraded by the cell. What we are left with is this is what is being degraded. So we have this short strand so this is the part that gets dissociated from this complex. So we have this short strand of mRNA molecule along with cleavage stimulatory factor, cleavage factor 1 and cleavage factor 2. So all of them get degraded. So now what we have is an mRNA molecule where the 3' tail has been truncated, it has been removed, but it remains associated with cleavage and polyadenylation factor. So this remains bound at the upstream poly A signal, and PAP also remains bound to the 3' end. Now this PAP slowly starts adding the poly A tail. So it adds the first 12 residues, and the addition of these 12 residues is comparatively slower. It's pretty slow, in fact. But later, PAP is joined by poly A binding protein 2. Binding of the poly A binding protein 2 to these residues, to the poly A tail, to the first 12 A residues, binding of the poly A binding protein 2 leads to rapid polyadenylation. So the first 12 A residues are added slowly, and they are added by PAP, poly A polymerase, and then poly A binding protein 2 binds to this poly A tail, and it increases the speed of polyadenylation. This is quickly up to 12 residues, and then another molecule of poly A binding protein 2 binds to it, and then this quickly adds another 12 molecules. So molecules are being added by the poly A polymerase, PAP, while poly A binding protein basically is doing two functions, is performing two functions. It is speeding up the addition of poly A tails by poly A polymerase, and it also controls the length of the poly A tail. So when it reaches roughly 200 to 250 residues, the poly A binding protein 2 stops PAP and does not allow PAP to add any residues anymore. So this is what leads to the formation of a mature mRNA molecule where you have 5' cap, and then you have got the coding sequences. After the introns have been removed, we will study RNA splicing later, and then we have polyadenylation. So till now we have talked about capping at the 5' end in details, and polyadenylation at the 3' end in details. So what else we need to talk about is there are two types of these poly A binding proteins. One is poly A binding protein C, PABPC. So C stands for cytoplasm, and we have this N1. So N1 is the poly A binding protein N1. I have mentioned it in the text on this slide. This is PABC and PABPN1. So N1 is basically the nuclear poly A binding protein. I have told you previously that mRNA molecule remains associated with lots of different proteins when they are found inside the nucleus, or even when they are exported out of the nucleus into the cytoplasm, they remain bound with multiple different proteins. So we have two types of poly-A binding proteins: those which remain associated with the mRNA molecule to the poly-A tail in this region. So we are particularly talking about the poly-A tail. So to the tail region, we have two types of proteins that remain associated: those which are found inside the nucleus and remain attached to the poly-A tail inside the nucleus. We call them poly-A binding protein N1. And then when this mRNA molecule is exported from the nucleus into the cell, then a different type of poly-A binding protein, what we call poly-A binding protein C, PABPC. So cytoplasmic poly-A binding protein that remains bound to the poly-A tail, and this plays a role in the translation process of the mRNA molecule. So I think that is sufficient for today. Thank you very much for your attention.